Great. So we'll get started with part 2 of this mass spectrometry and omics lecture. Today, to begin with, we'll discuss some of the examples regarding how metabolomics, for example, has really advanced our knowledge of biology beyond what we already know, like how it was really crucial to push the boundary. And then we'll discuss two other most widely used omics technologies. One is lipidomics, another is proteomics. And we'll also see at least one example each, that how they have really advanced our knowledge beyond what we know already from textbook. So, talking about metabolomics, how it has advanced the understanding of the biochemistry of biology that we knew earlier. Here, for example, is the famous case of isocitrate dehydrogenase 1 mutation, where instead of generating isocitrate to alpha ketoglutarate, it goes on, due to the mutation, it goes on to produce 2-hydroxyglutarate in abundance. And that 2-hydroxyglutarate, now only we understand that why in that mutation so many different changes, including changes in epigenomic landscape, etc., was taking place. The reason is that 2-hydroxyglutarate is a competitive inhibitor of these alpha ketoglutarate dependent dioxygenases that are involved in removing the methylation mark on DNA or RNA or even on proteins. So that's exactly why there was hypermethylation and repression of gene expression, which would otherwise really reduce the proliferation of these cancer cells. So in present, due to this production of 2-hydroxyglutarate, so this is a single mutant. It's a single metabolite, and it has been dubbed as an oncometabolite now. It can promote the tumorigenesis by changing the epigenetic landscape, as well as moving the redox balance. So here is one example we already discussed yesterday, the production of succinyl ion in case of humoral. And on the left-hand side, we can see the constitutive activation of NRF2, gain of function of NRF2, which is often observed in many aggressive type of cancers. NRF2 in general is known to be a protective gene. It's a good gene that we need to fight off oxidative stress. However, it was seen that cancer cells somehow has constitutive expression of this NRF2 gene, and that somehow probably helped them. And we could only understand how it did when we looked into the metabolism, and we did, in this case, we did both steady state as well as stable isotope leveling experiments, where we looked that it is basically a significant upregulation of pentose phosphate pathway. And what is happening that not only it is producing precursors for nucleic acid biosynthesis, but also it is increasing the production of NADPH. And when NADPH production is up, that gives these cells an advantage not only towards increasing lipid biosynthesis, lipid which is required for synthesis of new membranes, of new cells, new cancer cells that will be generated, but also for energy storage. And also by increasing NADPH, it is tilting the redox balance towards a reducing environment, and that makes more glutathione, reduced form of glutathione now available. And if you have reduced form of glutathione more available, that also helps in conjugation of reduction of reactive oxygen species, therefore reducing toxicity in these cancer cells, but also it increases the excretion of several drugs which are excreted often by glutathione conjugation. So it is just by increasing this pentose phosphate pathway flux, which is a metabolic event, what is happening here, that it is not only helping the cells to proliferate, but also it is allowing them to reduce toxicity from both reactive oxygen species as well as drugs by helping drug reflux. And this is another example which tells us that our knowledge of so-called, what is the biochemical landscape and what are the enzymes involved in which pathways are quite in. As we can see, lactate dehydrogenase that can produce 2-hydroxyglutarate, and this was identified by stabilization as well as steady-state experiments. So our knowledge of so-called metabolome as well as this whole biochemical landscape has to probably undergo significant expansion and modification. And one of that expansion will come from identification of not only novel metabolic activity, which we just discussed a couple of slides, but also identification of new metabolites. Okay, for example, here what we showed here that in this case these esters of dicarboxylic acid with either diol or propanediol. These are the molecules. It was found that when there is an increase in dioxin-induced toxicity, these molecules are hydrolyzed because these molecules are rather not hydrolyzed because the CES3, the esterase expression goes down, and therefore there is an increase in abundance of this molecule, and this increase in abundance of these CES3 substrates, which we identified first time, it is an indicator which pops up even before there is any significant change in typical, liver enzyme profiles. So that means this metabolic event is a much earlier event and which can act as a biomarker for hepatotoxicity, at least in this case in mouse models. And we proved that in several different mouse models of NASH. And then on the left side, you can see this molecule here, which is ribofluoroacyl creatine, D-ribofluoroacyl creatine. This is a novel sugar and amine conjugate, and we found that this goes up significantly. First, we found it in lung cancer, but also we later on found it in several different other types of cancer. However, unlike in case of the CES3 substrates, we do not right now understand what is the biochemical origin, and as we discussed, that is one of the challenges in metabolomics and pushing our knowledge in biochemistry. And down here, you can see another molecule also, which was found to go up. This is taurosebacic acid conjugate between decarboxylic acid and taurine, which is a novel metabolite that we identified. So it is quite incomplete. There are many, many more molecules and many, many more different biochemical pathways that are waiting to be discovered. And then on right-hand side, you can see two examples where we discussed about how gut flora has significant influence on our physiology. And in this case, as you can see, that this metabolite, which is a metabolite of choline, which is produced by the gut flora, the TMA, then further gets oxidized to give TMAO or trimethylamine oxide. So these type of metabolites are called co-metabolite because they are partly produced by our own enzymatic system, endogenous enzymes, and partly produced by enzymes from other organisms which are cohabitant in this case in the gut microbes. So this metabolite has been shown that is very important in driving inflammation and atherosclerosis and cardiovascular disease. So again, how this choline is basically derived from fat. If you take a lot of fat and stuffs like that, eventually you will have, and if you have a unfavorable gut microbial biochemical activity, then you will have trimethylamine oxide, and that will reduce atherogenesis and increase CVD risk. Similarly, it was found that gut flora-related metabolite can also alter the bile acid metabolism and deconjugation, and thereby promote tumorigenesis. So as so far we could see that there is a really lot that we can identify. In all these cases, just to mention, mass spectrometry was used as the tool to identify all these compounds. So mass spectrometry is really handful. So now we will move into lipidomics, and the reason we jump from metabolomics to lipidomics is often the border between metabolomics and lipidomics is quite understandable, as we just saw an example that lipid metabolism and other metabolic pathways are intricately connected, and therefore oftentimes it is also difficult to segregate them into. Oftentimes, if you really need to understand metabolism in detail, then you have to look at lipids as well as non-lipids, which are either metabolites or precursors of lipids. So let us just see some of the precursors of lipids. Moiety in lipids can come from sugar, eventual fatty acid, glycerol, amino acid, etc. The propionyl group can come from valine, leucine, isoleucine, methionine, glyoxylate, dicarboxylate, origin fatty acid. And on the other hand, the isoprene units can be derived from acetyl CoA and glyceraldehyde 3-phosphate, and these isoprenes eventually go on to use, and also towards synthesis of sterols. So as you can see, these two are often very connected, and also when you actually process samples, many of these metabolites that we discussed earlier, as well as lipids, they come together. So there has to be a way, and here is this link to Lipid Library, and this is a great resource for learning about how to, extract and analyze lipids. You must, if you are interested in lipids, please go through that. Lipid map, lipid classification system, lipid maps, we'll talk about this list. Has been, because there are so many different diverse class of lipids, so there is a database that has been generated, lipid maps, and here. And this is also a great resource for understanding different types of lipids, how they are connected, not only structurally, but also biochemically. So if they are generally, they indicate fatty acids or fatty acids, for example, palmitic acid, stearic acid, arachidonic acid, and stuff like that. Glycerol lipids are indicated by GL, and so you have to have a glycerol component, and then you have to have a fatty acid chain connected to that. We'll have all the structures shortly, and then you can have glycerophospholipids, sphingolipids, and these are basically how they are named. And so this is, for example, how they look. Fatty acids, very simple molecule, and then they can be conjugated to either glycerol or phosphoglycerol, or it can be conjugated to choline. And then you have sphingolipids, where you have amine instead of a simple glycerol. And then you can have sterol lipids. So this is the sterol, and it can also be esterified with fatty acids. And then you have prenol lipids like this. Of the vitamins that we know of are prenol lipids. So all these things you can find out in lipid maps. So the question is that, okay, now we know that there are so many different types of lipids, how to go about their analysis. So first thing first is, of course, extraction. And these are different type of extraction that you can think of. So for example, you can have a biphasic extraction where you have a polar or aqueous phase and non-polar or organic phase. And for example, the Falch method, which is a very old method, you can see that uses chloroform and methanol. Then you can have Blyder, which introduces chloroform, methanol, and water, and dichloromethane methanol. Or recently, this MTB methanol water was used because MTB layer, typically in all other cases, the organic layer is at the bottom and aqueous layer at the top. So therefore, when you are trying to take out the lipid, so that lipid, that thing you have to, really push through the aqueous layer and then take the organic layer. In case of MTB, because this is a light, low density ether, so that ether layer floats over the aqueous layer. So therefore, it is very easy to take the organic layer out and process it. And also it is important to note that sometimes when you have the organic layer down, there can be a contamination of the aqueous stuff when you are trying to take them out and the other way around. But if you have the organic layer on top, and that's what you are interested in lipidomics, so it is very easy to take it out and without much of contamination. And also there are other methods, for example, monophagic method like simple acetone based extraction. And then there is a butanol based extraction, and it was shown particularly that this butanol based extraction gives really significant recovery of polar lipids, including cardiolipids and stuff like that. So it really depends on what you are interested in. And then oftentimes many of these methods you add either salt or acid to reduce the solubility of the fat into the aqueous layer and push them towards the organic layer. So it quite depends on what type of lipid you are interested in. And as we have emphasized before as well, there is no single method that is going to give you every molecule, every type of molecule, and with 100% recovery. So therefore, you really need to optimize your method depending on what kind of analysis you want to do. If you want to go for, if you don't have any targeted set of compound that you want to look at, then you can simply start with something like Blyde method or MTB. That gives you a pretty good coverage to begin with. This is, for example, an analytical workflow. Let's say you have a cell or tissue, or you can have, serum or any other plasma. And you do, of course, if you have a cell or tissue, you have to either rupture or homogenize it. And then you do a biophagic extraction. And the aqueous layer will mostly have metabolites or very short chain fatty acids, which are highly organic acids, which are highly water soluble. So you can take that away from metabolomics. And in the organic layer, you will have mostly lipid stuff. And depending on what kind of lipid you want to look at, you would like to then subject them to different types of analysis. For example, if you really want to look at just the fatty acid composition, the best method probably GCMS-based fatty acid analysis by generating the fatty acid methyl esters. And you have to remember that when you are producing methyl esters, if you use something like an acid, methanolic HCl and stuff like that, it will not only esterify the free fatty acids, but also it will esterify the esterified fatty acid, that means the complex lipids. So you have to keep in mind that when you are looking into that, you are looking into the entire set of fatty acids, either conjugated or unconjugated. So if you want to look at just unconjugated fatty acid, for example, you can just do an MSTFA derivatization, silylation, that will not touch the esterified fatty acids. Okay, we will just look into free fatty acid. And then with the organic layer, you can also do reverse phase based ESIMS, electrospray mass spectrometry. That is most widely used. MALDI can also be used. You have to dry and spot it and you can use it, but because there is a significant interference from the matrix compounds, MALDI is not really widely used for looking into metabolites particularly, and also in case of lipids. However, very big lipids, you can have a reasonably good coverage from MALDI. In fact, MALDI-based imaging mass spectrometry analysis mostly throws up lipid spaces. And that's true for DESI-based and other imaging mass spectrometry as well. Many kinds of lipids, as you have seen, for example, the glycerophosphocholine kind of lipid is very easy to ionize. In fact, they have a resident charge on the trimethylamine at the end of choline. So therefore, it is very easy to get them ionized and analyzed as a positively charged star. Multivariate, and as we have discussed and we have mentioned yesterday, that once you have generated all these analytical data, whether it is from metabolites or proteins or peptides or lipids, once you have generated all this data, so this high dimensional data, then you do some multivariate data analysis to reduce dimensionality to find the pattern. We talked about PCA, PLSDA, OPLSDA, and several other, volcano plots and stuffs like that yesterday. So those are all methods that are still applicable to this kind of data set as well. So once you have that data, then you will, just like we showed yesterday, you will find out the features of interest, and then you will try to identify what are the features of interest. That means between the phenotypes that you are interested in, let's say phenotype A and phenotype B, sample set A and sample set B, what is that? What are those ions? First, you find out that had changed, and then you try to do the fragmentation analysis and find out, of course, using the help of the database, that what all these molecules may be. Okay, and eventually, as we discussed, that oftentimes at the end of the day, you would like to confirm that with authentic standards. However, that may always not be possible. But depending on the kind of fragmentation pattern that you get, if that is clear enough, and we'll discuss some of the methods by which you can do that analysis, you can be often very sure what the exact composition is. So, and never to forget that quality control samples and internal standards. As we discussed other day, your blank samples, your pooled samples, your cocktail of authentic standards that you must run intermittently to make sure that the instrument response is right. Also to make sure that you are able to take care of any variability that has been introduced because of sample handling. And there is always this issue of data normalization that we talked about, which you can do either by internal standards or by some endogenous compound, normalizing by some endogenous compound, although we don't have a very good one for lipidomics or metabolomics either. And then you can also do it by total ion count normalization and take care of any artifact that might be formed by your data. So those things are still valid as much as in metabolomics as in lipidomics. And then this is another schematics just to show you that what are the different kind of analysis that you can do and what kind of lipids you are. For example, if you start with a lipid extract and you find multiple negative ions in SIMS and you SIMSMS, you are probably going to look at most of the anionic lipids. And if you get a multiple positive ion and in your SIMS and MSMS spectra, you are probably looking into protonated or sodiated lipid and probably glycerophosphocholine or ceramide type of lipid. And then if you do a mildly basic lipid extract, addition of base, for example, LiOH, and again, you do multiple, you get multiple negative ion in your SIMS or MSMS, you are probably looking into weakly anionic lipids. And on the other hand, if you see most of the positive ions, then they are probably neutral but polar lipids, okay? Like saccharolipids, they can be neutral but polar. So, and then you can do chemical derivatization, and depending on the functional group present on the lipid, they will give you specific lipid classes and specific type of, ionization pattern. This is, for example, an extract from, liver tissue that we analyzed. It's a mouse liver tissue, actually. And here you can see this is a reverse phase chromatography And here you can see the LPC, LPE, LPC are lysophosphocholine, LPE, lysophosphoethanolamine. You have all the names, full names written down there. So those molecules are smaller lipids, so they come first, and then you will have slightly bigger but polar lipids, and then you will have at the end triglyceride, which are the most non-polar and they are also non-charged. And you have to remember, just like in case of metabolomics, and then if you take ESI negative, you will first see fatty acid, LPCLP. However, fatty acid sensitivity is not typically great in case of ESI. Fatty acid analysis is much better if you do by GCMS. And then you will have phosphocholine, phosphoethanolamine, or phosphoserine, and phosphatidic acid, which are certainly negatively charged. Now, as I was saying, that as you have seen in case of metabolomics, similarly in case of lipidomics as well, there can be several types of adducts. Particularly, for example, if you have triglyceride, which are not easy to ionize, they often, or protonate, they often come out as something like a sodium adduct or something like an ammonium adduct. Okay, so even looking at those, you can actually get an idea that what kind of lipid class you are looking at. So this is another example of fatty acid methyl esters, which are analyzed in serum samples, and you can see several different types of fatty acids that are identified here, and of all as fatty acid methyl esters, and you can do a simple GCMS matching, like we have library and you can base your fragmentation pattern with the library reported electron impact ionization spectra, and then you can also match the retention time if you have authentic standard, and that would be the final nail on the coffin of identification. So as we discussed, that once you thus have analyzed all your lipid, will again you will get a chromatogram just as we have shown, and as we discussed earlier yesterday, that you will deconvolute them, and oftentimes, unlike in case of metabolomics, in lipidomics you will have many co-eluting or very closely eluting peaks. So a good deconvolution is extremely important. So you deconvolute them, and at the end of the day, you will get a data matrix which comprises of the mass retention time pair and the intensities for each sample. Okay, so with that, this is the you will see very similar to what you see in case of mix, and then you will again do those analysis that we discussed yesterday. I won't go again. Similarly, you will identify features that correspond to the type that you are interested in or the sample group that you are interested in. And then, unlike metabolite identification, in this case, you have lipid identification. So while identifying lipid, what you do, first thing that you try to do is to take help of databases. Okay, so there are quite a few databases that are available, and most useful of them is one is Lipid Maps, and then one is HMDB, also good for metabolites as we discussed, and then you have methylene. Case of methylene, HMDB, both of them, you have experimentally reported spectra whenever available. And in case of lipid maps, actually you can generate, spectral fragmentation pattern. So for example, this is because lipid maps is, exclusively for lipids, and this is what we will be discussing here. So here you have, in your data matrix that you have analyzed, you have come up with a set of, ions that you want to identify as what are. So therefore, and you can see there are all these different types of metabolites that are available, and you can choose which type of lipids that you want to search for. Okay, and then you can give your mass tolerance window, depending, that will depend on what kind of instrument that you have used, and you have to give the ion type, what type of ion you expect. For example, here we have chosen the protonation, as we have discussed, that it may not simply be protonation. It may also be sodium adduct or a neutral loss or ammonium adduct and stuffs like that. So you have to be mindful about that. And then you can specify whether you want to use only even chain lipids, or you want to also include odd chain ones, and then you can sort them by delta or the mass error that we are that is calculated by the database. So this is a set of lipids that is pasted here, and let's say this is the set of lipids that you found to be most significantly distinguishing between the sample sets or the phenotypes that you are interested in. So based on that, you paste that and get. So for each mass that you have given, you will get the matched mass along with the delta. That means the mass difference that is there. And it will give you what kind of molecule it is. For example, it can be ceramide or it can be LPC. As you can see, for a simple one mass that you have given, you can have several of them as your plausible hits. Okay, and it will also give you, and since you have, in this case, we are only selected as a protonated species, so of course it will show you as protonated species as well. If you select it to be something else, it will show up as that. So similarly for 520, you can have several other hits. Now the question is that, and so if you click on each of these hits, for example, you see here, so these are simple lipids where you have only one, for example, there is only one fatty acid chain. And but you can have more than one fatty acid chain connected to a backbone, or if it is a ceramide, so you can. So for example, this is a phosphatidic acid here, and the overall formula is 34.1. 34.1. That means there are 34 carbons and one unsaturated. And this is how lipids are identified. So and mentioned. So you have to write the lipid class, and in the bracket you have, say, for example, how many carbons and what is the there are nomenclatures that we will not get into, that how you exactly name the position of the hydrogen bond, the carbon-carbon double bond as well, the unsaturation which is present as well. So for example, if you look at here, if you just take this guy, the phosphatidic acid here, it can be these many different combinations that can give you the same mass. So this is really challenging. This is not a very trivial task to exactly identify what they are, just based on the accurate mass. As we have discussed, that there are very high resolution instruments. For example, we have sub-ppm level of mass accuracy if we look into the Orbitrap or FTICR MS. However, even with zero error, if you are trying to identify a lipid, this is what you will end up with, because all of them will have exactly the same formula and same mass. So then you have to you have to then, for example, here is a ceramide, and you can see this ceramide D44-2 can be these many different combinations. Okay, and of course you could choose while you are searching, if you for some reason assume that there is no question of getting any odd chain fatty acid here, which is often the case if you are particularly looking into the cell lines, etc. Then in that case, you may omit the odd chain fatty acids, and the list will be slightly reduced, but not you will still not get the exact ID, because you still have many even chain combinations that will give you the same mass. So then you have to, of course, depend on the fragmentation pattern. Okay. So before that, there are a few things that you can decide on. For example, if the molecular ion if it is m by z value, if it is even, okay, then it is PC, P, or PS. If it is odd, then it is either PI, PG, PA, or SM. Okay. So again, the exact name of these classes are given here. For example, PI is glycerophosphorinositol, and PG is glycerophosphoglycerol, and SM is sphingomyelin, and PA glycerophosphate or phosphatidic acid, and then that we have discussed. Okay. And then these are the different, if this is specifically talking about the SI analysis. So these are the different modes where you are most likely to encounter them. So if it is a even mass look that has popped up in positive as well as negative mode, then you are probably looking into something like PC or P. But the PAs, the phosphoserine stuffs, generally do not pop up in ESI positive. You will only see it in ESI negative, and so on and so forth. Again, sphingomyelin can be seen in both ESI positive and negative. Then, often these lipids, they have in-source fragmentation. That means that is because there are groups which are very easy to cleave up and give rise to an ion. For example, the phosphocholine head group 184. So that is a signature of phosphocholines, and wherever you have that phosphocholine precursor mass that you are looking for, right along with that, you will find a coeluting 184 peak as well as few other peaks that we have mentioned here. On the other hand, in negative ion mode, you will get 224 and 140 minus. And also, this will give you a neutral loss of 60 in negative ion mode. So looking into these coeluting fragments or neutral losses, you can guess very much that what kind of molecule you may be looking at. For example, if you have phosphocholine or sphingomyelin, you can see, first of all, one will have even mass, another will have odd mass, although 184 will be present in both. So therefore, looking at 184 and whether the precursor mass is even or odd, you can distinguish between whether it is phosphocholine or sphingomyelin. Okay. So similarly, phosphatidic acid, you will see in negative mode, and 153 minus and 135 minus, and so on and so forth. So phosphoserines, typically, you can pick up very easily using the fact that, see, for example, 153 is present in both phosphatidic acid and phosphoserine in negative mode. However, phosphoserine will have even mass only, phosphatidic acid will have odd mass only, and then phosphoserine will have neutral loss of 185 in positive mode and 87. Okay. So looking into these signatures, you can make a very good guess. For example, this is important because in some cases you will see, for example, here that you have all kinds of hits. You have phosphoethanolamine, you have phosphocholine, and you have ceramide. So looking into the precursor mass and also the fragmentation. This is, for example, here we have given search in lipid maps for a mass of 480.31 and a 68.36. You can see that here there is in the negative ion mode, and here this is LPE 18.0 for sure and nothing else, because it gives you the fragment for the fatty acid 18.0. Similarly, the similar fragment you can also see in LPC. If you see in negative mode, as we have discussed, LPC also comes in negative mode as formic acid adduct, and there also you will see this. So therefore, looking for the fragments and the side chain, and here you can see for LPC that we discussed, the phosphocolines will give a neutral loss of 60 from the formic acid adduct. You can see here 568, it gives you a neutral loss of 60 to give you 508. So by looking into that pattern, you can see that these are both lipids containing fatty acid 18.0. And then you can always have, for example, here 78257, and if you search, this will come up as a phosphocholine 36.4 in the lipid maps, which is shown here. So 36.4, as we discussed, can be any combination. Okay, so different types of combinations are possible to give you PC 36.4. So one of the combinations, of course, this and this, but you will assign and confirm this by the presence of, first of all, phosphocholine is 184 in positive mode. We discussed that fragment, and you will also have the other smaller fragments here. And then you will have the LPC because of the loss of one fatty acyl side chain. And that LPC is basically telling you this is a 16.0 LPC because the loss is of exactly equivalent to corresponding to the loss of presence of a 24 side chain. On the other hand, for 18.2, the loss will be different, and looking at both the fragment which is present here as well as the difference between these two masses, you can identify what is the combination of fatty acid which is present there. However, this only tells you in most cases that what is the SN1 and SN2 position, but where these double bonds are located, and sometimes SN1, SN2 are basically the positions in glycerol where the fatty acid is connected. Okay, maybe like this. So this is SN1, SN2. These are so whether a fatty acid present in here or they are exchanged, you will guess reasonably based on the fragmentation pattern that we just discussed on that. And also there are several other techniques. However, the exact position of the double bond on that fatty acid side chain is something which is quite challenging to determine, because all this tells, the masses tells you that there are 4 unsaturations present, but that is something this is clueless about. So we will discuss some of the techniques that can help you to assign the now there is so far we have discussed that you analyze your data, you first record the full spectrum, and then you analyze the data, and then you choose which is the precursor mass that you are interested in, and then you target that mass, and using something like a triple quad or something like that, or a QTOF kind of instrument where you can do tandem mass spectrometry. You analyze the fragmentation pattern and try to infer what is the lipid species that you are looking at. However, that is very time consuming. So therefore, people have come up with this idea of shotgun lipidomics, and this is the same similar approach is also useful in proteomics as well, where and the reason this is so widely applicable in lipidomics and proteomics is that the fragmentation pattern in lipids and in case of proteins, therefore peptides, are relatively simpler or streamlined, I should say. Okay, it's something that you can theoretically predict. However, the fragmentation pattern for metabolites, because they are so diverse in nature, and you don't know what else you are looking at, as we have given some example where you have no clue, like there are different types of novel metabolites that may be present. So it becomes challenging to use shotgun approach in case of metabolomics. However, in principle, that could be done as well. So what you do in shotgun lipidomics, or for that matter, we'll also discuss shotgun proteomics. So here you basically, as you go on, scanning through the retention time window, and you have a M by G window that you are looking at, you fragment that M by G window in different intervals, and you keep on fragmenting anything and everything that comes in there. So there are two types of shotgun approaches there. One is data independent, one is data dependent, and another is data independent. The basic thing right now you can take, and we will discuss more about this. The basic thing is that in case of data dependent thing, your choice of what is that you want to fragment is based on what is that there in the spectra. So typically, you will choose the most intense peak that you have within that retention time window, and that's what you will fragment within whatever specified mass window that your instrument can select. On the other hand, in case of data independent approach, you try to fragment anything and everything that comes in your way. Basically, blindly, anything that is coming is getting fragmented, and you record all the data. So obviously, in case of data independent approach, the data size is much larger, and therefore the informatics load, the informatics challenge is also much larger. It's true for lipidomics as well as, and much challenging much more challenging in proteomics as well. So that's what you do. So basically, what you are doing, instead of waiting for the whole analysis to be complete and then allowing and then choosing which masses, which precursors to fragment and identify, you do that as you are actually running the experiment. And that has been possible because of the fact that now we have very fast scanning mass spectrometers, because within a given time window, your mass spectrometry cycle time has to be significantly shorter than the shortest peak that you can get in your chromatogram. Because anything there, the reason is that in case of, as we discussed, in case of lipidomics as well as I will see in proteomics, because they are so complex, you will have many different kinds of lipids coeluting or eluting very closely to each other. So therefore, within the same envelope, you have to really keep on scanning through the whole mass range and fragmenting them again and again. So typically, it is recommended that if you have a Gaussian peak, you will have at least 12 data points on a Gaussian peak, okay? If you want to get a good peak set. So your time window, therefore, has to be the retention times, the lowest retention time window or the peak width that you get by 12, less than that. So your scanning cycle, your mass spec scanning cycle, the whole selection of mass, fragmentation, analysis, and scanning through the whole mass region has to be very, uh, fast. And obviously, it is understandable if you are trying to, in case of data dependent analysis, you are trying to focus on typically the most intense peak. On the other hand, in data independent analysis, you will be looking into all the different types of peak, anything that comes. So therefore, you will have many more number of scans. So therefore, you have to have even faster scanning instrument. So, so this is again an example, and these are different classes of lipids and their ionization pattern and the mass to charge ratio ion that is typically used to monitor them. So after you have analyzed it, if you want to specifically look into a particular class of lipid, okay, so instead of, let's say, you just want to analyze how many different types of phosphocholine are there. So in that case, you can choose positive ionization mode and the 184 ion, or negative ionization mode and 168 ion. So in triple quad, for example, you will your third quad will be sitting at 184, and the first quad will be scanning and looking for anything and everything that is giving you 184. So that precursor mass will then show up, and you know that this is phosphocholine because it gave you the 184 ion in the third quad. Okay. So similarly, you can do others. So by doing this, you can simplify your analysis, and using the same sample, you can run the analysis again and again for different types of lipid species. Just a second. How we are doing by time? Okay, I have it's approximately. Okay, so we'll probably pace it up a little bit. Okay, so as we were discussing, that it is sometimes very challenging to identify what is the position of what is the fatty acyl chain which is present in SN1 or SN2 position, and what is the position of double bond. So in this case, as you can see, this is a high energy collision induced dissociation assisted with UV induced photo dissociation. In this case, using this technique, you can see that this particular lipid, which is, oops, this is particular lipid, both of them, they are having the same overall formula, which is PC34-1. However, in this case, and here the fatty acyl chains are also same, but the position of the fatty acyl chain are different. In this case, this is SN116, and in this case, this is SN1 18-1. Okay, so here you can see their fragmentation pattern is quite different, and by looking at here, you can see X3. In the zoomed scan, you can see that these two can give you clear idea that these two types of these two protein, lipids are different. So here again is another example where this species is 18-1 N12. That means this is the position, and this is N1. That means the position of double bond is different. And here also, you can see that looking into the fragmentation pattern, you can distinguish the position of the double bond. So the probably most robust and widely used method for assigning the position of a double bond is ozonolysis. And in this case, actually, In's group, they developed this ozonolysis protocol along with chromatography, where you can see that as this compound, this lipid undergoes ozonolysis. Ozonolysis will lead to cleavage of this bond and formation of a carbonyl compound, and that will lead to production of this 760, 650.4 ion in positive mode, of course, from 760, and they can monitor this by combining both mass spectrometry and chromatography. So here, for example, they have shown that how you can distinguish between this PC, not only the position of the double bond, this is even useful for analyzing the geometry of the double bond. That means whether it is cis or trans. Okay. Here, for example, you can see that this is both of them are PC 36-2, and therefore the same mass, and also having the same side chain, except for the fact that in this case, this was 2 trans double bonds, and in this case, this was 2 cis double bonds. And as we know that if you do ozonolysis, you can distinguish between cis and trans isomers. And looking into the fragmentation pattern, as you can see here, that these will clearly give you not only 2 different peaks in the chromatogram, but also 2 different spectra that you will get out of the products of ozonolysis. So looking at that, you can distinguish between, and you can assign the position of the double bond and distinguish between such isomeric compound. Recently, Feng et al., they have also used another method where they have used metachloroperbenzoic acid to do epoxidation and then collision-induced dissociation. And in this method also, they have shown that you can distinguish between PCs having different double bond position and assign their double bond position. And not only for PC, they have shown that it is applicable to fatty acids, LPs, LPCs, LPAs, and all different kinds of very versatile method that has been developed. So now, doing all this, what is that we, doing all this lipidomic analysis, all these LPCs, LPs, and all these things we have known for ages. So is there something different that, our ability to use mass spectrometry gives us to advance really lipidomics beyond the lipids and the biochemistry that we know? Yes, in this paper, which was published by Sarah Maria Fendt's group in Nature in 2019, they showed that SCD16, which is an enzyme involved in desaturating production of palmitoleic acid from palmitic acid, and palmitoleic acid is eventually used for membrane synthesis, and SCD16 is therefore very important enzyme. Biosynthetic enzyme, and cancer cells are expected to be dependent on that, because in order to produce new membrane species for new cells, they will require SCD activity. However, it was found that even when SCD activity was inhibited, then also these cells were somehow able to thrive. Eventually, they used mass spectrometry and found, and also they, of course, use molecular biology, including shRNA technology, RNA interference, to show that there is this enzyme, this FADase2, which actually produces an isomer of palmitoleic acid. So you can see these two here. This is palmitoleic acid, and here this is sapienic acid, and the difference is between the position of the double bond. So this is a different type of alternative desaturase activity, which is used by cancer cells to circumvent the SCD, the reduction or inhibition in SCD activity, and still go on with membrane lipid synthesis. So therefore, you can imagine that in many of the cases, people may be using SCD inhibitors as a way to reduce cancer cell proliferation and to treat cancer as a potential therapy. But in many cases, if there is this FADase activity, this alternative desaturase activity is present, then those patients are never going to respond adequately to the treatment. Okay, so therefore, this is again a new alternative pathway that we uncovered just using the power of mass spectrometry, and of course complementing it with other molecular biology techniques. And that is always true. It's mass spectrometry itself is not an answer to everything, but it gives you lead like where you should look into, what kind of thing you should do. Now we'll come to proteomics. So as we know, proteins are the executive agents, and we have approximately, let's say, 2,000 genes, and then from those genes we can have around 20,000 protein coding genes, I mean, and from them you can have different type of splicing and so on and so forth, and you can have approximately 1,000 transcripts. But the proteoforms, which constitute the protein, are more than 100,000, because there are several types of modifications using post-transcriptional modifications. And in addition to that, we have discussed in the initial class, there can be different types of, different forms of folded or partially folded or unfolded form of protein. And that can lead to a difference in activity over and above the fact that there can be chemical modifications of proteins and peptides. So how to identify them? We have talked about the structural aspect before, and now we'll be talking about the composition, rather, of the chemical composition and structure of the proteins and peptides. So how to go about that? So typically, there old school methods for looking into these molecular techniques have been chromatography techniques, ELISA, Western blotting, and stuff like that. And still, a lot of experiments are based on Western blots, and we know that not. So there has to be some method. And also, we have antibodies, good antibodies for only a handful of proteins, and they're expensive antibodies, they're expensive. So there has to be some method which can be applied irrespective of, even if I come up with a new protein today, I should be able to identify it. I should be able to identify what kind of post-translational modification it is there, and also how much of that protein or proteoform is there. So those things are often done using, let's say, protein microarray. And then there is gel-based approach like SDS page, 2D page, and 2D DIJ. And then, of course, we have advanced sequencing for identifying a protein sequence. And then we have mass spectrometry. So by mass spectrometry, we again have three kinds of questions in mind that we want to answer. First of all, who all are present there, and how much each of them are present, and how they interact with each other. The interact between proteins, etc., has been, I hope, discussed by Dr. Prasanna before, so we'll not get into that. So we'll mostly focus on how to identify the protein sequence, how to, or rather, how to identify the protein, and how to get to the post-translational modification, and then how to quantitate them. Okay, so these are typical steps. So this is just a cartoon where we have given cells or tissue. You can also have a biofluid, or you can have serum that you may have. The first step would be to extract them, and typically acetone precipitation is a reasonably good method for protein precipitation. And of course, if you have tissue, you have to first homogenize to release the protein, and then if you have biological fluids, however, you may simply use molecular weight cutoff membranes for biofluids. Now we know, for example, if you look at something like serum or plasma, there are a lot of very abundant proteins, including albumins, globulins, etc. So therefore, if you really want to, and if you are trying to, apply some sort of, analytical technique, some of them we'll discuss. Oftentimes, we will end up with, peptides which are originated from and mass spec signature, which originates from the highly abundant peptides. So oftentimes, while you are doing that, you have to deplete your abundant protein pool, and there are immunodepletion columns that are available, which can deplete these, let's say serum albumin, globulin, etc. And then once you have your so-called treated protein matrix, depleted and cleaned up, then you can do some sort of digestion and you can do desalting, and then that will produce peptide. Peptides you can do analyze by mass spectrometry, and so while doing that, we often combine that with some sort of chromatography technique, particularly in case of ESIMS. In case of MALDI also, you can use LC to spot the MALDI plate, and you can take different fractions from an LC and spot on different wells of the MALDI plate, and thereby do some separation. However, it is readily attachable to something like LC-ESI, and ESI is probably that technique which obviously has taken over, particularly while looking into the whole proteome global analysis, the discovery platform. MALDI is relatively limited in use nowadays, mostly for analyzing known proteins or relatively simpler protein mixtures and post-translational identifications and so on and so forth. For really large-scale analysis, electrospray ionization is more useful, and there is also a difference between the ionization mechanism of MALDI and ESI. As we know, in case of MALDI, you will mostly get singly charged species. You get doubly or triply charged species, but they are generally very low. On the other hand, electrospray ionization, as we have discussed, can do multiple charging, and multiple charging has advantage in terms of fragmentation, which will, I hope I have one slide where we'll show an example how it can help to generate a more informative spectrum. And then once you have the data, you will do the signal processing and data analysis, which is, again, once you have the data matrix, is pretty similar to how you handle the metabolomic data, etc. However, given that there are different types of, the data load is very high, and several peptides are generated from the proteome. So this data is much more complex, and particularly in terms of the informatic treatment of the data is quite involving. However, it has been streamlined to quite extent. Doing noise. Okay. So we have around 20-25 minutes. I hope I'll be able to cover stuff. Okay. So just like we have discussed before, just in case of both metabolomics, lipidomics, and proteomics, as soon as possible in your processing step, you introduce an internal standard, and oftentimes that is a isotope label standard, because isotope label standard will have exactly same chromatographic pattern, exactly same ionization pattern. However, the mass will be different, and thereby you can distinguish it, but every other effect that it will face will be similar to the analyte of your interest, and therefore you will be able to take care of a lot of artifacts. So the earliest you can introduce an isotope label standard, it is better for getting a more precise and accurate data. So this is a seminal paper by Klesh Pandey and Matthias Mann, where they reviewed back in 2000, and that was approximately that. And analyze the proteome, and you can match it. And that's the time actually the genomics project was also data, and how you could combine advances in mass spectrometry and advance in the genomics technology in order to really expand the horizon of proteomics technology. So just to give it as a schematic, so you have, let's say, two conditions. So you extract and solubilize proteins from, let's say, these are cells, and then you can separate it by, some sort of, let's say, gel electrophoresis on one dimension, SDS page, and on another dimension, isoelectric focusing, which is dependent on the PI. And again, when you choose that, and this is called 2D gel, where one dimension is according to the molecular weight and another dimension is according to PI. So you can choose the PI strips, the IEF strips, and the PI range that you want to focus on. And depending on the PI range that is covered by the IEF strip, you can really relatively focus your data. Okay, and also depending on what kind of subcellular localization the protein you are extracting from, that also can really simplify your proteome, and you can get a really focused information. Because if you just homogenize everything, then all the organelles are gone and everything from every organelle is now jumped up together. But if you really want to get organelle resolved information, then it may be a better idea to really extract, individual organelle functions. Sorry, and choose a PI range accordingly. For example, if you are trying to look into the histones, which are mostly positively charged, and therefore the PI values will be somewhere up. Compared to that, if you have something which, set of protein which are very negatively charged, in that case, the PI values will be somewhere down. So depending on that, if you have a specific type of, or subset of proteome you want to analyze, you can choose your PI ranges accordingly. And once you have done that, you can reduce your protein. And so typically first you do the isoelectric focusing, and then you can reduce and alkylate. So reduction will make sure that all the thiol linkages are broken down, so you will get only and alkylated, so they will not form again. So you will only get monomers of stuff. So you are not going to get a lot of dimers, etc. And then you subject it to some sort of SDS page, and then you can stain it. You can have fluorescent dyes like Cy3, Cy5, etc. We'll not get into that. And then you will get certain spots, and you find out what are the differential spots. You pick them up and you subject them to mass spectrometry. Okay, and that can be done either by MALDI or SI. As we discussed, nowadays mostly SI is used, but because things are relatively simplified and separated here, gel-based proteomics can be done very well with MALDI still today. If you are interested in specific proteins, what is a protein and what is a... So here, let's say you have a gel stuff, and you cut the gel and you take the protein band out, you subject it to trypsin digestion. Instead of trypsin, you can use other types of proteases as well. In fact, that will help you sometime, particularly if you are looking into identification of something like a novel protein. The tryptip digest may not be good enough, and you may like to have orthogonal information or using different peptides which will cut like GLUT-C and all, which will cut at different positions. So different like serine proteases and all this type of thing. So you can use different type of proteases to generate different data set and confirm and convince yourself that you are actually getting the same sequence or hitting the same sequence that you are getting by all. So you are getting a consensus hit identification. So, as we said, once you have these tryptic digest, you can either analyze them by MALDI, or you can analyze them by ESI, and oftentimes it is connected to liquid chromatography. And sometimes you can also enrich those peptides by using different types of affinity chromatography like metal resin chromatography. So these are like titanium dioxide, or there can be iron oxide columns, which can enrich phosphate containing peptides. So by looking into them, you have a better chance of identifying even low level of phosphorylation of your particular protein that you are interested in. And and you can again do phosphatase treatment, etc., to confirm that the modification that you are suspecting is actually a phosphate modification or not. So there are different types of things that you can do depending on what you are interested in. So again, talking about a generic workflow, let's say this is a peptide, and as we have seen earlier, these peptides have these monoisotopic residue masses and corresponding ammonium ion, and we have discussed earlier how to calculate the residue mass of peptide, acid, and how to calculate the mass of a peptide, and how a peptide fragments. We have discussed all this before. So these are the same thing that will be used even when we are doing this process. So again, these are some of the products produced, and on top of that, of course, we have side chain fragmentations. So you have a tryptic peptide, and you take these peptide for aspect-based sequencing. So basically, you select the mass. Let's say it's a triple. So you select the mass in quad, and then you and the fragmentation will happen as we have discussed. The B and Y ions mostly will be generated if it is a collision-induced dissociation, and you can do a sequence work to see whether you are getting the sequence that it is supposed to be. However, as we will shortly show, that you have the bioinformatics complementation of it, by which you will significantly simplify this exercise. So while talking about fragmentation, because a lot of, whatever you are doing in proteomics, including identification of the peptide sequence and, in the peptide identity, getting the modification, etc., everything depends on what kind of fragments you get. Because the overall mass of the peptide will not give you any sequence. The sequence will come from the fact that how they are connected to each other, in which sequence, in which pattern. So, and what are the side chains, etc. So we have to remember that there are things that affect the peptide fragmentation pattern. One is the peptide N by Z value. If the peptide is very large, then it will require more energy to fragment. If it is smaller peptide, it will fragment easily. If it is higher charge state, so there is, it will be easy to fragment because the Coulombic repulsion will help. And if it is lower charge state, then it will be other way around. So that is the reason that if you have in MALDI, it is difficult to fragment peptides because they are mostly singly charged. On the other hand, in ESI, you get multiply charged species, therefore it is easy to fragment, and you get a much rich fragmentation spectra compared to what you get in ESI. Collision energy, of course, if you more collision energy, there will be more fragmentation pattern. Fragmentation includes also on collision gas and mass spectrometer. As we just talked about, the MALDI will have lower fragmentation compared to ESI, and ion trap versus Q-TOF will have different type of fragmentation pattern, and then mode of dissociation. Okay, there are different types of dissociations, for example, CID or ETD or ECD. So CID is typically for precursors for like smaller peptides less than 5 kDa. On the other hand, ETD, ECD can be used for higher ones as well. And of course, amino acid composition of the peptide, which we discussed. So for example, if we take this glucoalbumopeptide, this is the sequence here, and a low energy Q-TOF fragmentation of a W charge species will look like this. You can do this exercise for yourself. It is It would be fun, but it will take a long time before I take you through the sequencing of each of these. So you have basically this B ions. So this is the B ion series, and on in this B ions as well. Y ion series here. So here is the Y ion series from the same guy. So by confirming this, you can actually get reasonable idea like what is the sequence of this. So if you take the same peptide in the MALDI, you can see that this earlier spectra was much richer, whereas in MALDI, the fragments that you get are much because the singly charged species. On the other hand, the earlier one was charged state affects the one of the now when you do this sequence work, one of the challenges typically people people how to distinguish between isoleucine and leucine because they have the exact same mass and therefore the B and Y ions are not going to give you any information about whether it is isoleucine or leucine. So for that, you can choose the B or Y ion corresponding to that leucine or isoleucine containing peptide, and you can fragment it further, for example, as was done here. And only in case of we'll get a fragment which is at 69. If you have leucine, you will not get that. So therefore, if you find out that in your peptide sequence, I mean, if in your mass have the 16 ion peak, then you will know it is isoleucine. So by doing that, so that is the only amino acid which two amino acid which have exact same masses, and you can distinguish them by looking further into fragmenting further the overall, this is what you do. You have a protein, you do trypsin digestion, you get proteolytic peptides, you have a mass spec, and then, as we were saying, that at almost the same time, we have a huge, upsurge of information from genomics data. So we could get the information about all these different genes, and we could translate them to corresponding amino acid sequence. And now that we know that where the trypsin cuts basically on arginine or lysine. Therefore, we will know if you have theoretically a protein, a given protein, then what sort of trypsin digests you expect. So what will be the precursor masses, the overall, the peptide masses that you expect. So, and then if you do MS on them, and there you will get the B and Y ion series. So you have two levels of match. One level of match is based on the raw peptide mass. Another level match would be based on the B and Y ion series that you expect from theoretical fragmentation of these proteolytic products. So based on the matching of these, MS and MS signatures, you can score. So in principle, you can theoretically fragment all peptides, all protein coding, not peptides, all protein coding genes and corresponding proteins in silico. So you will have a library of different peptides that will be generated from each of these proteins, and you can theoretically generate the ions that you expect from these peptide according to the fragmentation pattern that we discussed. And then with that theoretical fragmentation pattern, you match your mass spectrum and try to see where it matches most. And based on that, you get a score. So this is what we do day in and day out when we do the what you need a peptide mass spectrum, which is generated experimentally. You need the protein, so which is from gene banks, which brought, etc. And you need a search engine that will match your experimental spectrum, match for your experimental spectra in the theoretically generated spectra from the protein database. So these are different protein databases, and these are different. And most widely used is GenBank or SwissProt. And most widely used. And for example, you can use. So again, so just to summarize what you do experimentally, you have a protein sample and at your you have your protein sequences from all protein coding genes, and you have a fragment sequence uh, with the mass and fragmentation pattern. Then you so this is, for example, the multi-top opportunity, and then you can convert this to extract the pick list, and these are the different masses which are present here. And then you submit these masses mascot. So here you will it will ask for different name, search title, etc., and then it will ask you what enzyme you have used, and of course what what is the taxonomy. Will choose the corresponding the protein. Once you mention which enzyme you are going to use, then it will fragment it accordingly. And you can also mention different types of modifications that might be present, like many a time we have biochemical protein and stuff like that. So those are different types of modifications that you can define, and then these are fixed modifications that you are doing intentionally. On the other hand, there can be variable modification that are that that may occur because of post-transfer modification like N-acetyl or oxidation of methionine and stuffs like that. So you can define those post-transfer modification. It will accordingly change the peptide mass and fragmentation pattern, and then you have your query set, and you also define your mass tolerance limit in ppm, stuffs like that, and eventually it and those uh, in this case we got a hit which is fine albumin, and this is the red ones are the matched peptide. These are the peptides, as you can see. This is so like the first one, it is cut after R, and then there is between you can see there are more lysines, and many a time we have to remember that there can be one or two, you know, um, one or two missing, cut sites, because it may be because of several reasons. It may be because the trypsin was not the best quality trypsin, you didn't incubate it enough, or the protein was not unfolded enough, because it will cut only unfolded protein. So if it is folded form, then it will not cut. So and that is one of the ways people do this limited proteolysis to really map, what are the different regions of protein which are folded or unfolded. So obviously, if you do not have properly unfolded protein, it will not cut throughout. And in any case, some sites, so for these several reasons, there may be missing cut sites. So in several cases, and you can define that while you are searching, that how many maximum missing cut sites you can allow. Okay, you can define that as 1 or 2 or something. So it will generate the theoretical in silico spectra accordingly. So now we have discussed, how to identify a protein based on what is the time now. Okay, try to quickly go through the rest. So, so there are two approaches. One is top down and one is bottom up. Top down is nothing, and we have mostly been talking about so far. That you have a set of protein, and then you separate those proteins, you pick up each of the proteins, and then you you run on a gel, and then you identify what is their what is the fragmentation pattern, and based on that you identify the protein. On the other hand, bottom up is without any bias to any protein. You extract the protein, and then you do digestion, and you totally chop everything up, and you get this complex mixture. And so it is, you have all peptides from all the proteins which are present there, all sorts of modified or non-modified peptide, and then you try to separate them by LC. And as you can imagine, this will be a hugely complex chromatogram, and there may be several things that will be coeluting or very closely eluting. And this is exactly why the shotgun approach was really required in order to do the bottom-up proteomics. And these are different shotgun approach that available. I'll talk briefly about one of them. So one is data-dependent acquisition, where you select a particular window of the retention time, and then as you scan across retention time, you try to pick a peptide depending on what are the peptide masses that you are getting. You try to select one, mostly the most intense peptide, and you fragment that and you get that data. Like that you go on doing. Okay, so this is dependent on what is present. So based on the so you define that what you should be picking up to fragment. On the other hand, data independent acquisition is that you just define that this is the retention time, the chromatogram should be fragmented into retention time window, like let's say 2 seconds in this case, in the example. And then within each of these 2 seconds, you try to find out what are all masses that may be present there. So you can define a mass range, let's say, 400 to, let's say, 1500 or something like that. And you keep fragmenting every mass that is present there, and you keep reiterating again and again and again. So in the process, irrespective of whether it is a very intense peptide, low peptide or not, so it will fragment anything and everything that is present in that chromatogram. So of course you will get a very complex data set. So for example, here where you have given a SWAT, and this is the technology that was that is called SWAT for it's very informative. So you can give this window of, let's say, 25 Dalton. So it will chop up everything within that 25 Dalton, then cross over to next 25. That it will do ample number of time. I mean, many number of times across the retention. And after that, you get a huge data set. What happens? So after that, you will get a huge data set from which you again do the same. You follow the same logic that we have followed before, that you get this experimental data set, and you have your theoretical data set where you have generated all theoretical fragmentation pattern. And based on the match, you then decide that what is the protein that may be there, and you map and you try to call the proteins based on the sequence coverage and the best match that you get. And on top of that, so that is about sequence. What about the post-translational modification? Now, post-translational modification theoretically is quite easy, because you know that at least for known post-translational modification, what will be the different type of difference in mass that you will expect. For example, if you have a phosphorylation, you will have a delta M, which is the mass increase of plus 78. For N-acetyl glucosamine, it will be plus 203. Methylation plus 14. If it is acylation, it will be mass of the acyl group minus 1, and glycosylation, and so on and so forth. Okay, so citrullynation, for example, arginine to citrullynation, protein citrullynation, which is often seen in autoantibodies. In those cases, you will have a mass difference of plus 1 because of the hydrolysis. So, however, you have to remember, because it is a very complex mixture, oftentimes these modifications can give rise to change in mass, which is very close to each other, but given the fractional mass may be different. For example, if you have a lysine, acetylation will add up mass of 42.01056. On the other hand, trimethylation will have 42 unit, but 42.04695. So by accurate mass measurement, you can distinguish what kind of modification it might be. On top of that, in this particular example, you have to remember that acetylation will leave NH-COCH3 group, which has to be charged. On the other hand, trimethylation itself will bear a charge. Therefore, the charge state of the peptide will be different, and therefore the M by G value itself will be different. And by looking at that itself, you may be able to distinguish whether it is acetylation or. So in addition to these, signatures in mass spectrometry, there are different other tricks that people use. For example, people can modify these. People can you can, in case of phosphoproteins, because phosphoproteomics is one of the interesting areas to look at because of their involvement in signaling. So you can convert them to biotin tags. And so this is, for example, this is how it happens. So you have the phosphorylated stuff, and then you can bind to the biotin tagged stuff, which is industrialized, and then you can purify, and then you can purify that on evidence column, and then you can abundance of that tag. Now that you have a specific tag which you can monitor by mass spectrometry, you can identify what is the level of where the phosphorylation has happened, and between treated and untreated, what is the level of. Now we'll go to the quantitative analysis. There are several of them. We'll discuss some of them quickly. So the major advantage of level free, there are two types of quantitation. One is level free. That means you do not attach any tag. This is very cost and time effective. This is applicable to any and every protein. So this is proteome wide, and this gives a very high sensitivity. On the other hand, in case of level-based approaches, which we'll discuss shortly, that they are expensive reagents, takes up significant time for preparation. However, the precision and accuracy is very high, and the confidence in peak identification is very high. So there are different types of labeling techniques. One can be metabolic labeling, which is SELECT. Another can be the spiking of isotope level standards, which is typical practice in both lipidomics, proteomics, or there can be chemical or enzymatic labeling. So these are different type of labeling technologies, and here are some of the strengths and weaknesses of each. Because of the want of time, I will skip reading those then yourselves. So this is, for example, SILA. What you do here, you feed cells, your experimental sets that you want to compare with light or heavy amino acid. So for example, here in this case, you can feed them with 12C arginine or 13C arginine, or you can feed 12C lysine or 13C lysine. And then, so eventually, the cells will incorporate the 13C in all of its protein. And even if you mix these proteins, one set of protein which will have all 13C arginine will be coming only from the second group. And so in this case, let's say undifferentiated myoblast, and the light isotopes will all be coming from differentiated myotubes. So the advantage here is that you are not processing these samples separately. You are going to mix them. So they are going to suffer the same kind of interference if there is any, or same kind of artifacts if there is any, all throughout. Their ionization pattern will be very similar. only thing that will differentiate them is the mass because of the presence of the 13C labeled arginine in those peptides. And as you know, that the trypsin will cut them after the arginine, and therefore, so each of them will have either a lysine or arginine, and therefore, lysine or arginine are the ones mostly used for silac. But in principle, you can use other ones as well, depending on what peptide, what sort of proteases you use. And then eventually you can quantitate the light isotope containing peptide and heavy isotope containing peptide using the triple quad, and you will get exactly very precise and very accurate presentation of difference in expression of the protein of proteins in your two cells. To cartoon that initially you have all 12C level stuff, and then you enrich them with 13C, and slowly where you feed 13C, all the proteins, as you keep on expanding the cells, they will be enriched with 13C, and 12C will almost vanish, and then one set will be left with 12C only, another 13C. You mix them and you analyze. Chemical labeling, it is based on the reactivity of the functional groups present on the peptide. And here, for example, you can have a thiol reactive group, or you can have a amine reactive group and stuff like that. So ICAT was the first one that was developed, and in this case, there are hydrogens which are replaced by deuterium. So there is a light one where there are all hydrogens. In case of deuterated stuff, it will differ by 8 mass units, but everything else will be same. Biotin, you can purify your proteins as well, and using a in column. And then this tag will help you to distinguish two peptides. So again, you mix them and you get the ratio of heavy and light, which will tell you the level of expression, difference in expression of proteins. In case of ITRAC, you have this kind of a moiety, and here these places are where these are the places where you can have 4 different 13C, and depending on, or you can also change 15N. So by adding whether you have 2 13C, 1 13C, or 13C plus 15N, you can have 4 different masses. And here you can also use 4 different masses by combining isotopes of 13C. There are 2 13C and 18O. So you can have 0 and 13C, 18O, and 13C plus 18O. So overall mass will be exactly same in all cases, which will be 145. But this reporter group will tell you which group you have taken from. So you label the proteins of, you can have 4 different treatment groups. You label the proteins with 4 different isobaric tag with 4 different reporter mass. And this is where it will react with the amine specific peptide reactive group. So amines will attach here like this. And N-hydroxysuccinimide is the moiety which is often used for labeling amine. And for 8-plex, you will have all these different positions that can be labeled with 13C along with 2 nitrogens, giving you 8 different possible combination of isobaric tag and reporter group. And you can compare 8 different groups for, again, you can mix them, and by looking into different reporter masses, you can find out TMT, is tandem mass tag, which is very similar in principle. And here you will have 6 different labeling positions, and you can compare 6 different groups. However, the problem is that if you really want to, for example, if you want to analyze a set of 1,000 samples, want to exactly identify what is the level of difference, I mean, what is the protein expression difference, then there is no 1,000 different tags. So in that case, you have to really depend on the ability of mass spectrometers to be, for example, the data dependent or data dependent independent acquisition methods, where you can look at a specific precursor mass and specific the daughter mass, and monitoring their monitoring the intensity of this the parent and daughter pair. You can exactly see, even without any labeling, you can do the level-free analysis of quantities, analysis of change in protein expression, and that is what is very useful, like SWAP and all is very useful for analyzing several different protein samples, and it is also very cost effective. So yeah, this is just the last slide. So just again to give you an example, so whether all these things have really helped us learn something new. Here is an example where a myokine, which is irisin, which was, so these are, uh, these are proteins that are released by muscle when we do exercise. And the presence of this protein was really disputed. The gene was known FNDC5, but there was a lot of dispute regarding whether this protein was there or whether this is really something physiologically relevant. Only using mass spectrometry, we could really, in 2015, identify and quantitate this in human samples. So with that, and if there is any question, please share them. If not, then we will tell me and I'll be more happy to answer them. Is there any question from anybody? You can type in the chat box as well. Or if you want to take your time, you can take your time and email me. I'll be more than happy to revert back. Okay, then thanks a lot, and thanks for your attention, and please feel free to send me questions. Thank you.